Our immune system can be trained in several different ways. That's what we'll be talking about in this video. So as we discuss the adaptive immune system, there are some terms that we should become familiar with. The first of these, pathogens. These are disease-causing organisms. These are the things that make you sick. They can be different types of bacteria or parasites or even viruses. Even though viruses aren't technically alive, they are pathogens. So disease-causing organisms or substances. Now, antigens. These are going to be the signature molecules that are found on cells. These are the substances that our immune system recognize and elicit an immune response. And so, the signature molecules on pathogens, those are antigens. But really, even the signature molecules on our own cells, they can also be considered antigens. Our immune system just recognizes them as being ours, and so doesn't trigger an immune response against them. Then lastly, antibodies. Antibodies are proteins that are produced by our own immune system, produced from B cells, which are a type of white blood cell, and they are found in the blood plasma. They attach to one particular kind of antigen, and they help to counter its effects. So if a pathogen is being marked by antibodies, it is more likely that pathogen will be destroyed by other cells within our immune system. The adaptive immune system is a specialized system that provides specific responses to antigens of pathogens. The adaptive immune system is acquired over time based on which antigens someone is exposed to. So this adaptive immune system, it's able to provide a specific response to pathogens, but it needs to be introduced to those pathogens over time. Our adaptive immune system has immunological memory. So in addition to being specific, the immune system can remember antigens and react more quickly to second exposures to infection. This rapid, specific response can result in immunity. Much of the activity of the adaptive immune system relates to the production of antibodies. Antibodies are molecules which recognize and bind to one specific antigen. Immunity is resistance to specific invaders. It is usually acquired by natural infection. However, vaccination is a way of introducing someone's body to the antigens that are produced by a pathogen, and in that way giving them a head start on preparing defenses in case they encounter that pathogen at some point in the future. Vaccinations trigger active immunity, which stimulates the body to defend itself. Passive immunity, on the other hand, is receiving pre-made antibodies. Passive immunity may provide some instant benefit, but there's no long-term benefit from receiving pre-made antibodies. The very first time you encounter an antigen, it is known as the primary immune response. This first exposure to an antigen starts a slow process in the body known as clonal selection. This is the primary immune response. And in this particular graph, the primary immune response is represented by the red line. And we see with the primary immune response, antibodies will be produced, but it's going to be almost about 2 weeks after first exposure. And the total amount of antibodies that are produced is comparatively low. However, that blue line represents our second or subsequent exposures to that same antigen. And we see with our secondary immune response, antibody production ramps up within a few days, and usually this is sufficient to prevent an individual from getting sick at all. So the primary immune response usually takes several days to activate the cells that recognize that specific antigen. During the primary immune response, it is very likely that an individual will get sick from an infection because the pathogen is allowed to grow with little resistance as the body is figuring out its response to that pathogen. However, subsequent exposure to the same antigen will result in our secondary immune response being produced much faster. So our secondary immune response is produced when the same antigen is encountered again. It is initiated much faster due to the increased presence of the specific memory cells. These are white blood cells that recognize and remember that antigen. Clonal selection is this process of priming our body's defenses for repeat exposure to the same antigen. The body is not constantly producing large amounts of all antibodies at all times. That would be wasteful and inefficient. Instead, clonal selection is the process by which the immune system can select which antibodies it needs to mass produce and make more of those, thereby being much more efficient. So we have millions and billions of different memory B cells within our body, each of them recognizing different types of antigens. But it's only when we're exposed to that particular antigen that that specific B cell is activated. It is then going to start multiplying, increasing in number. Many of those new cells will become plasma cells, which release large amounts of antibodies. But we're also going to end up with more copies of those memory cells, so that if we experience that antigen again, our body can start this antibody production process much more rapidly. Antibody molecules have a few related functions within our body. First off, they need to recognize and bind to a specific antigen, a specific signature molecule. By binding to it, it can help to counter its effects. The immune system stops infections by destroying antigen-bearing invaders. When we talk about vaccination, what we're doing is we're trying to trigger that primary immune response at a time when the body is not actually in danger, giving the body a chance to see and recognize this antigen and prepare defenses for it without the pathogen really being present. So modern vaccination does not usually introduce a pathogen into an individual's body, but instead it introduces the antigens or signature molecules. This exposure to the antigen allows the process of clonal selection to start without the risk of people getting sick from the pathogen. Modern vaccines may provide the antigen itself, or it may provide instructions for the cell to make the antigen. And this is the case with the most recent mRNA vaccines for COVID. In our last module, we learned that RNA is a temporary molecule with instructions for making a protein. And so what happens is that the RNA is introduced into our cells. The mRNA lasts only for a short period of time. During that time, the protein that it codes for is made, And in the case of the mRNA vaccines for COVID, it is the spike protein, which are those antigens or signature molecules for the coronavirus. That protein that's made only lasts for a short period of time as well. Yet during that time, our immune system learns what the antigens look like without being exposed to the pathogen. Our cells and our body is able to end up making the defenses that they will need. And so it prepares a future defense should the body be exposed to that antigen again. Now, the strength of that defense and the duration of that defense, that is variable. There are some diseases where we receive a vaccination once and that's sufficient. Some where we need boosters. Unfortunately, so far with the COVID vaccines, it seems like a booster a year or so might end up being required. We'll have to see how that technology develops. But know that our immune system is being reminded of what those antigens look like and are preparing defenses in case of exposure. Contagious diseases can impact several individuals in a population. We'll talk about this field of study in our next video.